an image like no other inspiring. Decades of research and debate. Thus, Shroud of Turin, that's a piece of history, and it has an ongoing puzzle. For science, the Shroud of Turin is a burial cloth that's imprinted with what many believe is the actual image of Jesus after his crucifixion. Now it's been debated for years, but the shroud is back in the headlines in a major way. Deep within the ancient fibers of the Turin Shroud, scientists uncovered something astonishing: DNA. But when they analyzed it, the research team was left utterly speechless. For millions of believers worldwide, the shroud is the burial cloth of Jesus Christ, the only physical trace of the resurrection, a silent witness of fifth gospel written not in ink, but in blood. Before we continue, we kindly ask for your support. A like and a subscription are the best way to show your appreciation, and we truly value it. To skeptics, however, the shroud tells a very different story. They see it as the most sophisticated and convincing medieval forgery ever created. Some claim Leonardo da Vinci may have made it. Others argue it was the work of an unknown master, deliberately crafted to deceive pilgrims yearning for miracles. For more than 600 years, the debate has raged. Faith on one side, reason on the other, and neither has ever backed down. But in the night, in the 21st century, the era of pure philosophy and speculation came to an end. Science entered the scene, cold, precise, and uncompromising. Genetics, high-energy physics, spectroscopy, forensics. Technology made possible what was once unimaginable. Researchers stopped seeing the shroud as a sacred icon. They examined it as a crime scene as evidence in an unsolved case as a biological hard drive, silently recording 2,000 years of history. They extracted and sequenced the DNA embedded in the fibers. They scanned every single thread with X-rays. They broke the blood stains down into molecules, atoms, and elemental signatures. They expected simple answers, traces of a single individual, or perhaps remnants of pigment left by a medieval artist. But what they found was far from simple. The genetic analyzers at the University of Padua and these physicists working in the sealed laboratories of ENA discovered results that left even the most experienced scientists shaken. The data didn't just question existing theories, it shattered them completely. Both skeptical explanations and traditional religious interpretations collapsed under the weight of the results. The shroud was no longer just fabric, it became a map tracing a journey that began 2,000 years ago. Record of suffering so precise it cannot be reproduced. And evidence of an intense burst of energy. One whose nature modern physics still cannot explain. This marks the beginning of the most extensive forensic investigation ever undertaken. We will follow trails invisible to the human eye, microscopic pollen grains, molecular residues, logical fingerprints, and step by step, we will uncover whose blood truly soaked these threads. And why does it seem to scream of torture spoken not in words, but in the language of biochemistry? We will examine the physics behind the image itself, an image that functions as a photographic negative, a hologram, and an X-ray all at once. We will also answer a question that stunned the scientific world. Why DNA analysis forced researchers to admit that this artifact could not have been created in Europe and that it bears witness to events that altered human history. To truly understand the shock of modern genetic findings, we must return to the very beginning of scientific shroud research, modern syndology, back to the moment when humanity first truly saw the face hidden within the shadows of the linen. That moment came on May 28, 1898, in Turin. A lawyer, city council member, and passionate amateur photographer named Sacandopia was granted rare permission by King Alberto I to photograph the relic during its public exhibition held in honor of the prince's wedding. At the time, photography was far from simple. It was slow, technical, and demanding. Pierre Chenet hauled a camera the size of a suitcase onto tall scaffolding inside the cathedral. He relied on powerful magnesium flashes and lamps to cut. Through the dim interior, he exposed 2 large glass plates, each measuring 50 by 60 centimeters. Late that night, alone in his home dark room illuminated only by the faint glow of a red lantern, Pia lowered one of the plates into a bath of developing chemicals. And then it happened. As the image began to emerge on the glass pier nearly dropped, the priceless negative in shock. What he saw felt unreal, almost supernatural. On the photographic negative, where light should turn dark and dark should turn light, there appeared not a faint, blurry stain like the one visible on this cloth itself, but a sharp, high contrast, astonishingly detailed, positive image. A face deep-set closed, eyes a broken nose, a mustache, and forked beard, bruising on the right cheek. For the first time, the figure hidden in the linen was no longer a shadow. It was a man. The face carried an expression that was majestic, calm, and commanding, yet it belonged to a man who had just endured unimaginable physical suffering. The impact was overwhelming. It wasn't just a surprise. It was a revolution in how people understood the shroud. A normal drawing, whether made with paint, charcoal, or even blood, behaves predictably when turned into a photographic negative. Light areas become dark. Dark areas become light. The result is distorted, unnatural. The face turns into a flat, mask-like image. But the shroud was different. When photographed in negative, it looked like a true black and white photograph of a real living person. That single fact changed everything. And to this day, it remains impossible to dismiss, no matter how skeptical or determined to debunk it someone may be. What we see on the cloth with the naked eye is not a normal image at all. It is already a negative. And that raises an impossible question. Who, in the Middle Ages, in the 10th or 11th century, understood the principles of photography? Who knew that light could be reversed to create a hidden image? The answer is simple. No one. Who could deliberately create a perfect negative image without any way to preview or verify the result? Photography would not exist for another 800 years. The human eye and brain are not biologically capable of perceiving them world in negative, let alone reproducing it with such precise gradations of light and shadow. This was the first true mystery, this first crack in the seemingly solid wall of skepticism. The shroud did not behave like a painting. It behaved like a photographic plate, capturing a single moment in time. Over the years, the cloth has been examined with X-rays, ultraviolet light, infrared imaging, and laser scanning. Yet the greatest secret was never in the image itself. It was in the dirt, in the dust, in the microscopic particles trapped between the linen fibers during 2,000 years of movement, handling, and exposure. In 2015, a team of geneticists and biologists led by Professor Gianni Barcacci of the University of Padua was granted unprecedented access to the relic. Their mission was as bold as simple as it was unsettling to locate and analyze DNA. They were not searching for the DNA of God. Science doesn't know what such a trace should look like. There is no reference sample, no standard to compare it against. The researchers weren't trying to identify a single individual. They wanted to reconstruct the history of the cloth itself, where it had been, who had touched it, and whose hands had held it across centuries. To do this, they used specially designed sterile micro-vacuum devices fitted with ultrafine filters. These tools gently collected microscopic particles of dust, pollen, and organic material, not only from the surface of the fabric, but from the deep spaces between the warp and weft threads where ancient traces can remain sealed for millennia. They also analyzed particles gathered during the 1988 restoration, which had been carefully preserved in archival storage. This work pushed the limits of what science could safely attempt. Even the smallest modern contamination, a sneeze, a breath, a single flake of skin from a priest, a conservator, or technician could erase ancient signals and replace them with modern genetic noise. The samples were transported to an ultra-clean room-grade laboratory. There, the sequencing began using next-generation sequencing, or NGS. The scientists focused on mitochondrial DNA from both plants and humans, and for a crucial reason. Unlike nuclear DNA, mitochondrial DNA exists in hundreds of copies per cell, is passed down exclusively through the maternal line, and survives far longer in ancient material. It serves as a powerful and reliable marker of geographic origin and human migration. For weeks, computers ran non-stop, decoding millions of nucleotide sequences, comparing them against global genomic databases, representing populations from across the world. When the final diagrams appeared on the screen maps of genetic HLO groups and ancestral lineages, the researchers realized they were looking at something that defied any simple explanation or forgery. This was not the genetic profile of one person. It was a genetic portrait of humanity itself. The findings were published in the highly respected journal Nature Scientific Reports, and the reaction was explosive. The results sent shockwaves through the scientific community. The scientists expected to find one clear dominant genetic signature. If the shroud were a medieval forgery created in France, as skeptics have long argued, then European DNA, specifically French or Italian, should have overwhelmingly dominated the results. If, on the other hand, it were an authentic relic from Jerusalem that had never left the region, then the genetic traces should have pointed almost exclusively to the Middle East. That is not what they saw. What appeared instead was chaos. The entire world written onto a single piece of cloth. The shroud revealed genetic fingerprints from people across vast regions of Eurasia and Africa. Let's examine what that found. First, the Middle East. Scientists identified haplogroups commonly associated with the Druze, a tightly knit, historically isolated ethno-religious community living in the mountainous regions of Israel, Jordan, Lebanon, and Syria. Druze DNA is considered extremely ancient and remarkably stable, having changed little over thousands of years due to centuries of isolation. This makes it a powerful marker of geographic origin and provides strong evidence of a Middle Eastern connection. Second, Western Europe. Haplogroups such as U5B and H1 to H3 appeared exactly as expected. Since the 15th century, the shroud has been kept in Europe, first in Chambéry, then in Turin. It was handled by the Poor Clares nuns, who repaired it by members of the House of Savoy, who owned it, and by countless European pilgrims over the centuries. Third, North and East Africa. Haplogroup L3C M1A1 emerged, pointing to regions such as Egypt and Ethiopia. This was unexpected and deeply intriguing, suggesting contact with some of the world's earliest Christian communities in Africa. Fourth, South Asia. Haplogroups M39, M56, and R8 appeared, genetic markers typical of the Indian subcontinent. And most astonishing of all were traces from East Asia. Haplogroups such as D4 and G2A markers associated with China were present. China, India, Africa, Europe, the Middle East, all of it encoded into one ancient cloth. How could this be possible? If the shroud were a forgery, created in the damp basement of a French abbey around the year 1350, how could traces of Chinese and Indian DNA possibly be present on it? In the medieval world, globalization did not exist. There were long distance contacts, yes, but not on a scale vast enough to leave such widespread, clearly identifiable genetic traces on a single piece of cloth. The answer turned out to be even more remarkable than the mystery itself. The shroud is not merely a burial cloth. It is a traveler. When scientists in Padua mapped the genetic data, they made a stunning discovery. The genetic distribution aligned almost perfectly with precise geographic accuracy, with an ancient historical route that many scholars had long dismissed as legend. The journey of the man-made image, known as the Mandalion. According to early Byzantine, Syrian, and Arabic sources, the shroud was folded in four so that only the face was visible, displayed within a special frame. This folded relic, called the tetradiplolon, did not remain in Europe. It moved across the ancient world. The journey began in Jerusalem, the place of death and resurrection. From there it traveled to Edessa in the 2nd century. The shroud remained there for centuries, hidden within the city walls before being rediscovered. Edessa stood at a vital crossroads of the ancient world, the Great Silk Road. Caravans from China, India, Persia, and Arabia passed through the city. Merchants carrying silk and spices. Pilgrims, diplomats all came to venerate the relic believed to protect the city. They breathed near it. They kissed its casing. They touched the reliquary. And with each encounter, microscopic traces, skin cells, hair fragments, sweat settled onto the cloth. Layer upon layer, century after century, the DNA of the world accumulated on its surface like invisible dust. The journey continued. Third Constantinople. From 944 to 124, the relic was held in the heart of the Byzantine Empire. The emperors of Byzantium acquired it by purchase or by force and carried it to what was then the capital of the world. Constantinople was a true mega-city of the ancient world, a place where peoples of every race, culture, and region came together. Then in 1204, during the chaos of the Fourth Crusade, the city was violently looted. In the aftermath, the Shroud vanished. Next came Athens and Greece around 1205. After the fall of Constantinople, the relic passed into the hands of French knights and was kept in Athens for a period of time. Then France, Larry 1353. This marks the Shroud's first clearly documented appearance in Western Europe in the possession of the knight Geoffroy de Chauny. And this is where the genetic evidence becomes devastating to the medieval forgery theory. A forger working in the 15th century Europe could not possibly have gathered dust and DNA from people in China, India, the Middle East, and Africa, regions he knew only through distant travelers' tales like those of Marco Polo. Nor could he have deliberately planted those traces onto a cloth in a way that would deceive genetic scientists 600 years later. The DNA found on the Shroud is not random contamination. It is the collective, biological memory of humanity. It is dust from the sandals, robes, and hands of countless people who gazed upon this face over thousands of years. This biological record proves the antiquity of the object and its Eastern origin more convincingly than any radiocarbon test ever could. But human DNA is only half the story. The other half is written in plants. It's on palynology. The study of pollen offered another crucial piece of the puzzle. Two respected experts approached the mystery independently. Professor Anom Dan, a renowned Israeli botanist from the Hebrew University of Jerusalem, and the Swiss forensic scientist Max Frey, one of the pioneers of forensic pollen analysis. Working separately, they examined pollen grains trapped deep within the linen's fibers. Using adhesive tapes, they carefully lifted microscopic samples from the fabric. What they discovered caused a sensation in the botanical community. Pollen from 58 different plant species was identified on the Shroud. 17 of those species are native to Europe. Exactly what one would expect given the Shroud's documented history over the past 6 centuries. But the majority came from elsewhere. Plants native to the Middle East, Turkey, and the Anatolian steppe, perfectly matching the ancient route through Edessa and Constantinople. Then came the most striking discovery: a group of plants that grow nowhere else on Earth. Species found only in a narrow geographic corridor between Jerusalem and Jericho. At the center of this discovery was one plant in particular, Gandelia torn, a thorny desert shrub, a type of thistle. Its pollen accounted for nearly half of all the samples found on the cloth, an enormous and highly unusual concentration. Why would pollen from a thorny plant be so dominant? Why would thorns be associated with the burial cloth of a man treated as noble? The answer emerged when historians and theologians revisited the descriptions of Christ's passion. The crown of thorns, Gundelia tourniforti, with its long, rigid, needle-like thorns, is precisely the kind of plant Roman soldiers could have used to twist together a mocking crown for the king of the Jews. And suddenly the pollen began to speak. This plant blooms around Jerusalem at a very specific time, early spring, during the Jewish festival of Passover Pesach. The discovery of an unusually large concentration of pollen from this exact thistle concentrated around the head and shoulder areas of the cloth serves as direct evidence that the body was crowned with it. A second botanical clue came from Zygopilum domosum. This plant is endemic to a very limited region, growing only in the Judean desert and parts of the Sinai Peninsula. Its pollen was also found on the fabric in significant amounts. No medieval forger working in France could have obtained pollen from plants native only to Israel and then applied it invisibly at a microscopic level onto a piece of cloth. A forger would have used paint. Pollen cannot be painted. It acts like an invisible seal from the crime scene, a form of geographic fingerprint that cannot be forged. For many years, especially during the highly rational 19th century, skeptics argued that the reddish marks on the Shroud were nothing more than pigments, ochre cinnabar, or tempera, mixed with gelatin. That claim collapsed in 2017. A team of Italian researchers led by Professor Julio Fonti of the University of Padua, working alongside physicians from a hospital in Trieste, examined the stains using transmission electron microscopy and Raman spectroscopy. By analyzing the material at the nanoscale, they saw not pigment, but blood, human blood group AB. It is one of the rarest blood types, yet it appears frequently on ancient Christian relics, including the Sudarium of Oviedo. But this was not the blood of a healthy person. Within it, scientists identified nanoparticles of creatinine and ferritin bound to hemoglobin. Such extreme concentrations appear only under one condition: severe fatal trauma. Prolonged torture accompanied by dehydration and massive muscle damage. When muscle tissue is destroyed by repeated blows, a process known as rhabdomyolysis creatinine floods the bloodstream in enormous quantities, and the blood on the cloth recorded exactly that. This is a biochemical scream of pain. The analysis revealed that the man wrapped in this cloth did not simply die. He was beaten to a condition that was already incompatible with life before the crucifixion even began. This aligns perfectly with the gospel accounts of the Roman scourging carried out with flea, leather whips embedded with lead weights. The body shows evidence of more than 100 blows. The chemistry of the blood tells a story of suffering that no paintbrush could ever reproduce. An artist can paint the appearance of a wound, but no artist can imitate the biochemical signature of a body in extreme trauma, polytrauma, kidney failure, and hypovolemic shock. Another long-standing mystery was the color of the blood. The stains on the cloth appear red. Normally, ancient blood turns dark brown or black over time, but scientific analysis revealed unusually high levels of bilirubin, a substance released by the liver during extreme stress and severe trauma. Bilirubin preserves the red coloration of blood for centuries. The blood of a tortured man remains red. This is not mysticism. It is biochemistry under extreme stress. At this point, many ask the same question. What about the famous radiocarbon dating? In 1988, radiocarbon testing appeared to settle the debate once and for all. 3 of the world's leading laboratories, Oxford, Zurich, and Arizona, announced their results with 95% confidence. According to their measurements, the fabric dated to between 1260 and 1390, the Middle Ages of forgery. The world accepted the verdict. Even the church stepped back, referring to the shroud as an icon rather than a relic. But science never stops evolving. 30 years later, researchers identified where the fatal mistake had been made. The error was not in the technology. It was in the human choice of sample. In 1988, a tiny piece of cloth, no larger than a postage stamp, was cut from the very edge of the shroud. That corner had been handled countless times over the centuries, grasped by bishops and cardinals during public displays. Sweat, skin oils, candle wax, bacteria. That edge suffered more contamination and wear than any other part of the cloth. Further research by chemist Ray Rogers of Los Alamos revealed something even more damaging. That corner had been expertly repaired during the Middle Ages so skillfully that the repair went unnoticed. The sample was not representative of the shroud as a whole. And with that realization, the certainty of 1988 began to unravel. The French monks who restored the shroud did not use a modern technique known as invisible mending. Instead, they reinforced the damaged edge by weaving in new cotton threads, carefully dyed, to match the color of the linen. Later chemical analysis of the very sample cut and tested in 1988 revealed the presence of cotton fibers. That alone was a red flag. The main body of the shroud contains no cotton at all. It is made entirely of linen. To disguise the repair, the cotton threads were colored using alizarin dye to blend with the older fabric, and gum arabic was applied as a binding agent. In other words, the material tested in 1988 was not original cloth. The laboratories dated the patch, not the shroud itself. They measured the age of medieval cotton and centuries of accumulated grime, not the first century linen. This was not a minor oversight. It was a massive sampling error and a clear violation of basic archaeological protocol. Dating the most contaminated and heavily repaired section of an artifact is like trying to determine the age of an ancient statue by analyzing the chewing gums stuck to its base. Recognizing this flaw, scientists began searching for a method that could bypass contamination entirely. In 2022, Italian physicist Liberato Dero from the Institute of Crystallography in Bari applied a fundamentally new approach, one that does not rely on organic contamination at all. The technique is called wide-angle X-ray scattering, or WAXS, and it changed everything. This method focuses on the aging of the linen itself, specifically the cellulose within its fibers, measured at the atomic level. Over time, linen naturally changes. The long polymer chains of cellulose slowly break apart. Its crystalline structure degrades under constant exposure to background radiation, humidity, and temperature. This process acts as the material's internal clock. Darrow compared samples from the shroud with fabrics whose ages are firmly established, ranging from linen used to wrap Egyptian mummies dating back to 3000 BCC to medieval textiles from the 10th through the 14th centuries. The results stunned researchers. The aging curve of the shroud cellulose did not align with medieval fabrics at all. It was far older. In fact, the molecular structure of the shroud's linen matched almost perfectly with remarkable precision. Linen fragments recovered from the fortress of Masada in Israel. Masada fell in 74 AD, and the textiles found there are dated to between 50 and 74 AD, the 1st century, the time of Christ. Using advanced X-ray techniques, scientists pushed the age of the Turin Shroud back over 1,000 years, placing its origin in the heart of biblical history. In 1988, science seemed to dismiss the miracle. In 2022, with modern tools, it was reignited. But one question remains. How did the image form? There are no brushstrokes, no pigments, no ink. The image sits on the surface of the linen only 200 nanome deep, hundreds of times thinner than a human hair. Scrape it, and it vanishes. It is not paint. It is a chemical transformation caused by oxidation and dehydration, a burn left by an unknown source of energy. Scientists tried everything. Acid heat gamma rays, but only a short, intense pulse of vacuum ultraviolet radiation could replicate it. Yet such lasers did not exist in antiquity. To form the image across 4 square meters, the body would have had to emit an unimaginable burst of energy lasting less than a billionth of a second, enough to imprint the cloth, but not destroy it. In 1976, NASA technology revealed something even more incredible. The shroud contains a perfect anatomically accurate 3D image. The intensity of the imprint corresponds exactly to the distance between the body and the cloth, something no artist ancient or modern could reproduce. Even more astounding digital analysis showed coins on the eyes matching rare leper minted by Pontius Pilate in 29 AD. The probability that a medieval forger could have known this zero. The shroud also carries dust from Jerusalem pollen from plants that bloom only near the city in spring, and blood showing biochemical markers of extreme torture. The nails pierce the wrists, not the palms, showing precise forensic accuracy, including nerve reflexes in the thumbs. Layer by layer, discipline by discipline. Biology, chemistry as physics, geology, history, numismatics. The evidence points to Jerusalem 30 to 33 AD. The shroud defies explanation. It is not a painting. It is a holographic imprint of a real human being. A forensic record. A witness to a moment that changed history. Returned to its vault, the shroud remains silent. But the data speaks. Legend, myth, and faith are supported by measurable scientific evidence. Perhaps one day we will understand the flash of energy that left this trace. Until then, it stands as the 5th gospel, an eloquent witness to a world-changing event. Do you believe science will ever explain a miracle, or are some doors meant to remain open to the heart?